I'm here to talk about biocatalysis in the synthesis of valuable substances with cyanobacterial cells. And before I start, I would like to thank the organizing committee for giving me the opportunity to present some of my research work in this conference. So, however, my research is not um, quite is similar to what our colleague here, Philip, talked about, although he uses fungi cells, but I'm talking about cyanobacterial cells. How you can use cyanobacterial cells to catalyze chemical reactions and synthesize valuable substances from substrate. So first, I would like to talk about cyanobacterial cell biocatalyst. So cyanobacterial cells are blue-green algae that require simple nutrients for its cultivation and also requires carbon dioxide, water, and solar energy for biomass production. And according to research, for billions of years, cyanobacterial cells have been known as the first oxygen-producing photosynthetic microbes. So the reason why these cells were selected is because, one, it requires simple nutrients for their growth and cultivation. And secondly, their genomic structures are quite easy to work with, easy manipulation, and also they are of high environmental friendly. So selection of substrate in this research, I try to use the following substrate: one, phenyl acetate and phosphonic compounds. Although the phosphonic compounds I use dimethyl um, dimethyl methyl vinyl phosphonate and also diethyl ethyl vinyl phosphonate. So the other one I also used was epoxy phosphonate. So biocatalysis processes and synthesis of valuable compounds. This process actually involves it's a light dependent process. So, and it's been done in a growth chamber where the substrate and the cyanobacterial cells as biocatalysts are allowed to interact and leading to the production of additional high added value compound. So here, okay, all right, okay. So these are the material and methods used. So first we have microorganisms. So these microorganisms we selected as biocatalysts. Why we selected these microorganisms is because previous investigation revealed that these microorganisms have high potential, or they are active towards several substrates. And here we use Synechococcus species, Nostoc species, Kamptonema species, Limnospira species, and the Leptolyngbya species. So this BG1 and BG10, these are the media for the cultivation of these cyanobacterial cells. So this media, the difference between BG10 and BG11 is because BG10 does not have nitrate, because some cyanobacterial cells require nitrates for its cultivation, like BG11. So BG11, Synechococcus bigranulatus, growth in this in the growth medium for this Synechococcus species required nitrate for its cultivation. Why for the Nostoc, nitrate is not really suitable for the growth or for biomass production. So the next one is the starting material, like the substrate. Like I said before, the idea behind this is to use simple and inexpensive starting material and see how the robust capability of cyanobacterial cells to catalyze the reaction, leading to the production of high added value compounds. So here, like I said earlier, I use dimethyl vinyl phosphonate, dimethyl vinyl phosphonate, and also epoxy dimethyl phosphonate. Then finally, one phenyl ethyl acetate. So here we try to examine the possibility of cyanobacterial cells to carry out hydrolysis. Like I said before, light is also one of the most important things for the reaction to occur, because the photobiocatalytic reaction between the substrate and the biocatalyst is strictly light dependent, because light provide enough allow the reactants and the product and the reactants, as the biocatalyst and the substrate, to interact, giving them and enable them to reach enough activation energy, thereby speeding up the reaction processes. So the procedure of biotransformation, I try to summarize it here, but briefly I will explain. Media preparation, like I said earlier, we use BG10, BG11, and spirulina media. So the media was prepared according to the laboratory instructions, and the cyanobacterial cells were growth using, we have manually growth chamber and then the automatic growth chamber. So the manual growth chamber, we try to adjust the process parameter, the light, and also and suitable environmental favorable environmental condition necessary for the growth of the cyanobacterial cells. So then the photobiocatalytic reactions, the substrate and the biocatalyst were allowed to interact. Then after the interaction, for example, we this substrate and the biocatalyst allowed to interact within 2 hours, 4 hours, 6 hours, 8 hours interval. And we also try to use to extend the time by doing it for the period of 1 day, 3 days, 5 days, and 7 days. So after the whole process of a photobiocatalytic reaction, we try to use the centrifugation method to separate the biomasses from the products. So, and then the extraction process. The extraction process is simply we use the extraction solvent, ethyl acetate, to extract the product from the supernatant previously from the centrifugation process. Then analysis, these are the analytical machines used to determine the products of our biotransformation processes. We use liquid chromatography mass detector, mass spectroscopy detector. We use nuclear magnetic resonance of isotope one carbon 13, phosphorus NMR, 2D NMR, and then infrared spectroscopy and gas chromatography. So here in this figure, I try to summarize the whole process of this photobiocatalytic process. The first one, like I said, dimethyl vinyl phosphonate, which is the substrate. The biocatalysts used here are Limnospira maxima and Limnospira indica, and we use 21 days culture of these cyanobacterial cells. And the outcome of this biotransformation is of this reaction is biodegradation. So this in my next slide, I'm going to show you a table containing how this occurred. So then the figure 1b shows epoxy methyl dimethyl phosphonate is a substrate, and then we use Synechococcus granulatus shows a positive uh, outcome. That is the reason why we try to bring out Synechococcus granulatus. However, this experiment was also repeated using Limnospira maxima, Limnospira indica, and Kamptonema species. So and then the outcome of this, after a series of analysis, we realize that the product is dimethyl 3-hydroxy 1-ene phosphonate. This is a possible product. And then finally, figure 1c is 1-phenyl ethyl acetate. So here, the duration of biotransformation process here is 24 hours. However, we try to shorten the duration of biotransformation process to 2 days, 4 days, 6 days, and 8 days. However, the production of because this reaction is led to the production of R and S isomers of 1-phenyl ethanol. So we try to find the duration of time allow for the substrate and the biocatalyst to occur, leading to the production of enough enantiomeric excess. So we shortened the duration of biotransformation to 2 days, 4 days, 6 days. However, the result we got was we have limited enantiomeric excess. So the one with the maximum enantiomeric excess was the interaction that occur within 24 hours of biotransformation. And the cells used, the biocatalysts used here are Synechococcus species and Leptolyngbya species. So the next one, this is the phosphorus animal spectrum of the starting material, which is the epoxy methyl dimethyl phosphonate. So here, if you look at this figure very well, we have around here we have around 29.603. That's where the signal started from, and then here we have next we see after the biotransformation process, we got something like this. So the signal is around 22.15 here. And however, this other little signal you are saying is from the substrate. If I should go back, yeah, it's all about it's from the substrate, because here you see this same signal is still there because the substrate is not 100% pure. So this is because of that's why we see some of this little signal here. So then the next one is I tried to uh, analyze the results more to find out what exactly we have after the biotransformation of epoxy phosphonate. The reason why we selected this epoxy phosphonate and 1-phenyl ethyl acetate is because of these compounds are of high commercial importance. For example, 1-phenyl ethyl acetate could be used as precursors in food and cosmetic industries due to the rose-like flavor, and also goes to phosphonic epoxy phosphonate and vinyl phosphonate. They are really important in the pharmaceutical, cosmetic, and also cosmetic industries. So here, like I try to summarize, let me try to summarize this IR spectrum of the obtained product of epoxy phosphonate biotransformation. So if you look here very well, this broad signal here, it shows the presence of OH group, and how here we have the sharp signal around this place. It shows the presence of OH group. And that, sorry, okay, OH group is this one broad signal here, and this one sharp signal here shows the presence of CH group. And then this other one here you see is the PO group, as phosphonate group. So here, this figure was interpreted based on the information gotten from the literature, organic structure from Spectra, 4th edition. So the next one, we try to compare this the result we got after the biotransformation process with the starting the standard, which is epoxy methyl dimethyl phosphonate. So if you look very well, there is a clear distinction between the obtained product and the substrate. So the next one is, okay, like I said previously, in my previous slide, I said I will try to summarize the biodegradation process of vinyl phosphonate biotransformation process. Here, if you look here very well, this K animale means Kamptonema animale, and Kamptonema animale Limnospira indica are the biocatalysts used. So the growth media for this are BG11, BG10, and spirulina media. So for this, we try to check if this substrates are are stable in this media. So after 1 to 7 days and also to 30 days, we realize that the substrates are are, s- are stable in the growth media. So there was no biotransformation and there was nothing like biodegradation of the substrate. However, we try to uh, for the biocatalyst also, I also try to check using several analytical devices for if we can also get something like after 7 days or 21 days culture also, but we realized that there was nothing. So there is actually an interaction between the substrate and the biocatalyst. So DEVP is diethyl vinyl phosphonate. So for that ethyl vinyl phosphonate, 1 days, 3 days, 5 days, 7 days, and up to 30 days, there was nothing like bio transformation, not like biodegradation. However, dimethyl vinyl phosphonates in cooperation with the biocatalyst Limnospira indica here, uh, after 3 days, there was biodegradation. Up to 7 days, there was complete biodegradation of this substrate. And also for Limnospira indica with dimethyl vinyl phosphonate, the biodegradation starts from day 1, then until 30 days. So these are the structures of the substrate. The next one is the GC analysis report of 1-phenyl ethyl acetate bioconversion. So here, for figure 1A is the chromatogram of the standard starting material, 1-phenyl ethyl acetate. So that is, and also the chromatogram of the standard R and S 1-phenyl ethanol, and also the chromatogram after after 24 hours of biotransformation processes, we realize something like this. R and S phenyl ethanol as a product, having the bioconversion degree is 77%, and also the enantiomeric excess is 48%. However, this experiment is going to be repeated for longer period, maybe for like um, 1 day, 2 days, 3 days, to check if we can have enough of these enantiomeric excesses. Then also it goes to this one also. We have the complete biotransformation also occur here after 24 hours of interaction between the substrate and the biocatalyst here. However, we have a limited enantiomerase excess using Synechococcus granulatus. Leptolyngbya species shows a very positive result. So in conclusion, I would like to conclude that chiral 1-phenyl ethanol can be obtained through biotransformation of 1-phenyl ethyl acetate, which is a substance used in food and cosmetic industry due to its rose-like flavor. And the optically R and S phenyl acetate ethanol are the two most important ethanol used in pharmaceutical and cosmetic industries. So if you, like I said in my previous slide, biodegradation also were seen in vinyl phosphonate bioconversion process, and this demonstrate the potential of selected substrates to degrade some phosphonates and using them as source of nutrients. And obviously, we can see that the finding from this research is very important, as it limits or eliminates undesirable phosphonic compounds such as dimethyl vinyl phosphonate in the environment. And the reagent methyl 3-hydroxy in phosphonate obtained after the biotransformation of epoxy-methyl dimethyl phosphonate is still under investigation to examine its scientific and economic importance. So, and after the whole process, we are going to try to use the biomasses obtained from the biotransformation process to check if this biomass could accumulate rare earth metals in the next research, which is going to be done in cooperation with the Czech Republic, Czech Academy of Sciences in Czech Republic. Yeah. Thank you so much for your attention. These are my references, and thank you so much for your attention.